tell a little bit about who she is. And I would say, Amber, please take it away.
All right. Well, thank you so much for having me today. I am planning to sort of go through and talk about sort of how I came to where I am and sort of career things, and then go through about 30 minutes or so some recent research that I'm doing, and then talk about go into a little bit more detail on the analysis. Now, this class is probably going to be the least amount of math you're going to have in this class, because I'll mostly be talking about some pretty straightforward, pretty basic time series analysis. But I think it's still useful to talk about and go over and see how it's used in real life. So before I get into all of that, here is who I am and a little bit the career path I took. So I actually went to the University of Minnesota and had my bachelor's in neuroscience then, more when I was a clean slate, and then started to like build up a lot more in different types of neuro things. I always really enjoyed neuroscience. When I actually started at the University of Minnesota, I was actually in the chemical engineering program. But I knew I always wanted to be a physician and was really interested in the brain. And I essentially had an advisor tell me, well, why are you wasting a good engineering career on going into medical school? So that was, I probably shouldn't have listened to that, because I actually wish that I would have done more math and engineering. And that's partially why when I went to the University of Chicago and joined the MSTP program, I decided to do computational neuroscience instead of just plain neuroscience. Now, my background is quite a bit different than Mark's, who'll be talking more to you next week. I definitely came into computational neuroscience from more of a biological background and understanding with, just a little math. And I really appreciated the education, sort of learning more about the basics, especially when it comes to applied things like signals analysis and being able to represent neurons with systems of differential equations. But you all probably at this point know more math than I do now. I've forgotten a lot of it since it's been 20 years, but the principles are still there, and I'm really glad that I learned that. And yes, I was VIM's first graduate student about a little less than 20 years ago, and I took this class, I believe, around 19 years ago. So, and here are my peers. Mark, who'll be talking to you next week, and Jen, who we all were in VIM's lab together when I started and did our PhDs in VIM's lab, and were part of MD PhD programs in different ways. And then after I did my 8 years in the University of Chicago, getting the MD and the PhD, I went to San Francisco to do anatomic pathology residency and a neuropathology fellowship. From there, I was trying to figure out what I would do with the rest of my life. Did I still want to do research? Did I want to just do clinical work? And ultimately, I was exposed to someone who was starting to run a lot of clinical research looking at traumatic brain injury. And they thought this would be a really good area for a neuropathologist interested in research to go into. I went to a National Neurotrauma Society meeting, and there was all sorts of different research presentations, some that were focusing more on electrophysiology and circuits, which is my background, and I thought that was, very cool. And then on the other side, there was a lot of interest in looking at chronic traumatic encephalopathy and neurodegenerative pathologies that occur after traumatic brain injury. So I thought this area seems like a good way to sort of match my background in more circuits and electrophysiology with pathology. So that's how I ended up doing research in traumatic brain injury. And it turns out there's a lot of people interested in having a research neuropathologist who studies traumatic brain injury. And I ended up getting a job here at the University of Washington, where I'm a neuropathologist, and I do 20% clinical work and 80% research. Oh, and please interrupt me at any point in time if you have questions. This is really supposed to be an informal presentation and fun. So day to day, what I do now, 20% of my time is really spent predominantly looking at brain tumors and the pathology and diagnosing them with our current techniques. And, sometimes these are muscle biopsies, sometimes these are brain biopsies, sometimes spinal cord, so all those types of things. And then the rest of the time, the 80%, I have a mix of my research as part of running the brain bank here in the neuropathology core at the University of Washington. So I am one of the neuropathologists along with two others right now. And we work closely with a lot of different studies. There's the Alzheimer's Disease Research Center, who has people who donate their brains after they've been studied clinically during life. That we have the adult changes in thought study, which is an aging study in the community, where we sort of look at how people age and what happens with that. And then there's at least 10 more different studies that have been around in our center where we look at motor neuron diseases, we look at Parkinson's disease patients. And then why I ended up coming here is because about 5 years ago, one of the prior fellows and Dirk Keen, who sort of runs our neuropathology division, became really interested in traumatic brain injury. And I think this actually all started when I've heard the stories. Paul Allen, who used to own the Seahawks, when all this stuff came out about CTE, he became really concerned about it. And since he has the Allen Institute and has put a ton of money into neuroscience research, he came and talked to the head of neurosurgery here, who's like, we really need to do something about this and study this more. So then the chief of neurosurgery got the neuropathologists involved, and that's sort of how the very beginnings of how they started doing more traumatic brain injury research here. Well, that's expanded. They started by looking at studies in sort of our aging community and trying to look at the pathology of those who had TBI in the past and those who didn't. And they realized we really need more than just recruiting people who are good and healthy when they're 65 and trying to understand what TBI is doing to the brain. So we've now worked with our community and our medical examiners, and we've expanded our brain bank to look at community traumatic brain injury by working with the community. And that's really what I focus a lot of my research studies in the brain bank and human tissue on. But I look at a lot of neurodegenerative disease too. So that's part of it. And then the other part is I realized when I finished my clinical training, I really enjoyed electrophysiology and I just couldn't get away from it. So I got back into patching again after being away from it for like a decade. And that's become the other half of my research, is building my own lab and doing electrophysiology, where predominantly we're looking at mouse models of traumatic brain injury. But we are starting to get into human iPSC cultured neurons and looking at relationships between microglia and neurons. So today I'll be focusing most of my talk on the research I'm doing, predominantly more on this basic research side. But if you have any questions at all, please feel free to ask. So here are the two projects I'm going to be going into a little bit more detail about. I originally, of course, started in Vin's lab, really interested in neurons and oscillations and networks, and I still am, but I've become really interested in how neuroinflammation in the brain can change how neurons and networks work. And so I'm going to be talking to you today specifically about microglia and some of the changes that we're seeing after traumatic brain injury and how we think they might be working, or we actually sometimes don't even know that yet, but we know they're important in some of the results. So I'm going to be presenting that to you today. So I'm really focused on understanding predominantly the chronic effects of traumatic brain injury, and my research sort of focuses predominantly on these three main things. How does a traumatic brain injury eventually lead to neurodegenerative disease in some people decades later? How do we get a post-traumatic epilepsy, which often arises years later after traumatic brain injury? And how does it lead to dysfunction in the brain that can be progressive? And so a lot of the things that I'm doing are aimed at looking at those three questions, and probably these are all interrelated in some way. Now, the first project, I'm really going to be focusing on the orbital frontal cortex in the brain. And you might ask, well, why there to start? And of course, ideally, we will be looking at more and more areas in the brain, but I think the orbital frontal cortex is really important because it's one of the vulnerable areas in traumatic brain injury, despite the wide heterogeneity of mechanisms and severity of TBI. So on the left is just a picture from one of the neurosurgeons at UCSF that I used to work with, just showing where the most common locations of contusions are. And you can see really the orbital frontal cortex is really the epicenter where a lot of the contusions occur, as well as the anterior temporal lobes. And this is because of sort of the ridges in the skull. When the brain moves in the head, those ridges in the skull are there, and the brain just runs into it, producing contusions. And that's considered sort of more moderate to severe traumatic brain injury, depending on how you define it. But we're finding that even with people who have mild TBI, where they don't have any deficits on their imaging, any known lesions on MRI or CT by clinical images right now, we are still seeing that this area is showing some deficits. So this is just a track TBI study. This is the study that enrolls people who come into the emergency room with traumatic brain injury at 18 different sites across the country. In this particular study, they're looking at the ones in San Francisco, and they took these cases that had nothing on imaging. They're considered mild TBI, which means they at most were confused. They used the Glasgow Coma score to sort of rate that, and looked at their functional MRI about, I believe, 2 weeks after their injury, and they found that there was decreased connectivity, especially in those orbital frontal networks, despite them not having any lesions or anything like that at MRI. And this correlated with executive function score 6 months later. So I think this is a really important area to look at, not just with severe TBI where there's an obvious contusion, but in mild. And so there's a lot of questions on, well, what leads to chronic disease after TBI? And we have no idea why TBI is the risk factor for neurodegenerative disease. There's been lots of different mechanisms discussed, and here's sort of, I think, the two more prominent ones. On the left, the idea is, well, when you have axonal injury in the brain, you're changing the dynamics of the proteins that are binding to the microtubules. In this case, tau is really a microtubule stabilization protein. And then when you have axonal injury, you disrupt that, and then you start producing abnormal forms of tau that can eventually accumulate and turn into neurofibrillary tangles and act in a prion-like way to seed the brain and lead to neurodegenerative disease. And on the other hand, we have this idea that there's some type of prominent neuroinflammation that gets activated from the traumatic brain injury and never quite goes away. And this can be in the form of activated microglia or astrocytes that keep producing toxic substances or doing something to the neurons over time that just keeps leading to excitotoxicity and neuronal death that keeps progressing. So probably in real life, there's a lot of different things, and this is far too simplistic, but I decided, well, let's just start looking at this in actual human TBI. There hasn't really been many studies in human traumatic brain injury because this hasn't really been a focus of many brain banks until recently. I think outside of the University of Washington, Boston University, which is well known in the news for collecting the brains from a lot of football players, has what might be considered a traumatic brain injury brain bank. And then the Uniformed Services collects the brains of military veterans and active duty military. But that's all pretty recently. And so there hasn't, and the medical examiners usually, don't have the resources or the bandwidth to be doing research on these cases. And so a lot of what we know in humans is pretty little, surprisingly, compared to neurodegenerative disease. That work has been going along for decades plus now. So here's just a little bit of the basics of the brain donor cohort. When we have people who look at cases that come into the medical examiners, and then the medical examiner will ask the family if they're interested in brain donation. And if so, then someone will reach out and talk to them about our program, and then they might donate their brain to research. So it's a pretty stressful thing because a lot of times these are sudden deaths. So I'm always amazed at how willing families are to actually speak with us and to give the brains of their loved ones to science. So it's a pretty great gift. Then we usually have the family answer a questionnaire that goes into a little bit of the details about their history. And the basic questions we have are, did they ever had injury, yes or no? And then we ask a little bit more about, what type of head injury it was, and if they had symptoms afterwards. And we know a little bit more. Usually they have like an open notes field with they'll tell us about hospitalizations, or they'll tell us about medical records that we could get. So in this particular study, I decided to take control cases and age and sex match them across mild and severe TBI cases. In this case, mild TBI is defined as, yes, they had a head injury, and yes, they had symptoms afterwards that they complained about. And that could be anything from having a sleep problem to mood instability to concentration problems that lasted for weeks afterwards. And then severe TBI is considered those where they had a hospitalization for several weeks. They were noted to be in a coma or have skull fractures or actual lesions on their brain. And here's just the cases that we have that we decided to look at. We were able to find 11, and a lot of our studies are limited and by controls because it's harder to get people to donate the brain when their family member was pretty healthy and normal. But here are all the cases. The average age is in the 50s, and I think this is a really interesting range to look at because we sort of want to know, well, what's happening in the brain before, you know, they're in their 60s, 70s, and 80s and have neurodegenerative disease or other disorders of aging. And I think this is why this study is pretty unique. So the first thing we did was just do very simple immunohistochemistry. And all we did was stain for some different neuroinflammatory markers. IBA1 stands for microglia and macrophages. GFAP stands for astrocytes. CD68 stands for lysosomes, and they are increased in activated microglia and macrophages. And then on the neurodegenerative side, we stained for phosphorylated tau protein. We took the section from the orbital frontal cortex, and we actually just used a program called Halo, which if you could do the same thing in ImageJ, it's just a little bit easier to handle huge, big slides in our Halo program to just look at the percent area of staining. So we took all the brown pixels of a certain intensity, and that's shown here in the red. And then we looked at how much of the cortex is staining with this protein. And this was actually all done by Corinne, who actually, I think, took this class last year or the year before and was a University of Chicago student who's in my lab now. And she does a lot of the quantitative imaging analysis and now is doing a lot of electrophysiology. So what became pretty clear is that the strongest signal we see in this chronic TBI, and most of the TBIs are, you know, years to decades before death, we saw that the microglia signal was really the strongest in terms of the p-values. And we saw significant changes in the percent area of staining with microglia. That's not to say that other things weren't showing a trend. Certainly there was a trend in the staining with the GFAP, and we did see a little bit more tau staining as well in the orbital frontal cortex. But we thought the microglia signal was quite interesting. And one thing we noticed when we were just looking at the slides is we started to see a lot of these microglia in brown here that were sitting next to these neurons. And you can tell they're neurons by their nuclear morphology. They're usually larger and ovoid, and they have nucleoli, and oftentimes you can see sort of the pyramidal cytoplasm around them for the pyramidal neurons. And we're like, well, that's interesting. A lot of these microglia seem to be sitting next to these neurons, and we have more of them after TBI. So what is going on in this situation? And we went on to do immunofluorescent staining and be able to do Z-stacks and look in 3D with the MRS program and confirm that we really have increased numbers of these satellite microglia that are sitting and wrapping around neurons. And we see that it's increased for sure in our severe TBI group, but even our mild TBI group, where we don't see any obvious differences easily, there is an increase in the number of satellite microglia. So here's just some more evaluation of, well, what are these satellite microglia? They've been described, you know, more than 100 years ago, but then people have sort of ignored them and not really looked too much at what they're doing, but they seem to be different than just the rest of microglia. Not only do they sit and wrap around neurons, but we see how they change after traumatic brain injury is also different. So when we look at the non-satellite microglia that are just out in the parenchyma, we see that they become they have much higher volumes after TBI. They sort of take on sort of thicker processes, as you can sort of see here, and more extensive, larger cell bodies. But the satellite microglia don't seem to have those same changes after TBI, even though we have more of them surrounding neurons. So we became really interested in trying to understand, well, what are these microglia? How are they different from other microglia? And I am very particularly interested in, well, what are they important in doing to neurons and networks? So to do that, we had to move to a mouse model. Human tissue is great for showing you interesting, I think, correlations, but to really get at mechanism, you can't do a whole lot with just human tissue, at least not easily. So we have a mouse model, traumatic brain injury, that reproduces damage to the orbital frontal cortex in a similar way, at least somewhat similar to what we see in humans. So these mice have a sort of contusion that's applied over the prefrontal cortex with a piston, and the orbital frontal cortex is down here. They don't have any direct contusion in the orbital frontal cortex or any noticeable loss of neurons when you just look at an H&E slide, but they do have prominent deficits in behavior when you look at different types of behavior. And the orbital frontal cortex on lesion studies has been shown to be really important in reversal learning behavior, which in mice is very similar to the Wisconsin card sorting test in humans. And the Wisconsin card sorting test is you're switching between showing people a card and having them actually read a word or tell you what color it is. And then you'll change the rule on them, and they have to like sort of pick up on that rule change. And that's what we do with mice. We have them dig for food in different textures and odors, and then we switch the rule about where the food is hidden. And the mice are able to learn the initial rule just fine, but then when you switch it on them, they take longer to learn the rule than control mice. So we use this mouse model to also look at the orbital frontal cortex, and we can do this in a transgenic mouse that specifically labels microglia so that we could actually patch neurons adjacent to microglia or away from microglia. So we first decided, well, do we see these same changes in satellite microglia? And we did. We found that similar to humans, we got an increase in neurons that have satellite microglia after TBI. So then we went on to look at what's happening with neurons that are sitting adjacent to them. And here's just some examples of neurons that are filled with biocytin in the pipette while we're recording. And here's just an example of a neuron that has a satellite microglia sitting on top of it in orange, and an example recording here, and then just a normal pyramidal neuron that doesn't have any satellite microglia next to it, and then an example recording here. And this was sort of just for fun. I didn't know if this would actually, we'd see obvious changes, but surprisingly, we noticed that when we recorded from the neurons adjacent to microglia, they had this reduced excitability, which we're defining as the ability to fire action potentials with increasing current injection. So there's a reduced number at increasing current injections, amplitudes. And then we also have a decrease in the sort of first firing frequency here that occurs with increasing current injection. And this correlates, with some changes in the action potential, such as the threshold is higher in these neurons that have the microglia sitting next to them, and after hyperpolarization, after the action potential is greater in amplitude. So then we decided, well, that's happening in the control condition. Our original hypothesis is, well, we're probably having more satellite microglia come in as a compensation to try to dampen, excessive hyperexcitability that we are seeing in the network. So we went and looked in traumatic brain injury at the same type of thing. And surprisingly, we found that this wasn't the case after traumatic brain injury. And this is 1.5 months later, so a nice chronic time point. We found that we no longer have this reduction in excitability. And So satellite microglia are no longer able to reduce the intrinsic excitability of neurons. And now we're really interested in trying to understand a little bit more why that is. Well, does the same thing happen in humans? That's one thing we are trying to verify. Before I jump into that, I mentioned that the network is hyperexcitable. Well, what does that mean? I'm still using the same technique I used in graduate school in Vim's lab to look at how networks produce oscillations. We apply 0 magnesium and 5 millimolar potassium solution, which leads to unblocking of the magnesium in the NMDA receptor pore. And over time, you start to get oscillatory activity in the slice when you're patching in the network. So this is just an example. I'm patched onto a layer 5 pyramidal neuron. We're trying to hold its baseline here around negative 65 millivolts, and then we're just looking at the input that comes in. And if you wait long enough, you'll start to see these synchronized inputs over time that are polysynaptic, and we use sort of a cutoff of 2 millivolts to see sort of when they start and when you start to get regular synaptic inputs coming in, and over time they'll get larger and larger and larger. And so what we've been using has our measure of hyperexcitability is when these first occur, when that onset time is. And we have found that in our TBI slices that come from mice that have had a TBI 1.5 months earlier, or that onset time is sooner than in our sham slices. Of course, I'm getting Corinne to now doing more analysis of this and look into more Fourier transforms and frequency analysis to see how the high and low frequencies change over time, but that's still in its infancy. And this is, sort of the data we've published looking at this network to date. So we thought, well, we want to try to look in our human tissues to see if we can have a correlate. Is change in excitability that we're seeing, and we can't really easily just take tissue and record from it for humans. Of course, we did that, and people do that nowadays by taking tissue from people who've had epilepsy resections. And certainly some of those cases might have had a chronic TBI, but that's really hard to get. And I do at some point plan to record from human tissue from some of our epilepsy surgeons, similar to what I did with them in graduate school. Excuse me. But until that happens, we're just using immunohistochemistry to try to look for a correlate. So CFOS is one of the activated that shows that neurons are activated in some way. People usually use it after some type of behavior, and then they immediately sack the mouse and then try to use that as a marker of, well, these neurons are important in this activity. We thought we'd just look at it and see, well, do we see any relationships between this CFOS activation and the network state in general and with satellite microglia. So here's just an example of our mouse sections here in the sham and TBI conditions, and then our same human section here in the sham and TBI conditions. And here's an example of and red is just a Nissl stain to highlight neurons. Green is our microglia, and the blue color here is the CFOS staining. So I think you can see pretty easily that after TBI, there seems to be in both the human and the mouse conditions, although the level of staining is a little different, that we are getting more activation of neurons that might correlate with an overall hyperexcitable network. And then we also tried to look at neurons adjacent to the microglia here, noted with a star. And we are starting to see maybe a little bit where the ones that are adjacent to microglia and control have less number of CFOS positive activation versus after TBI, that doesn't seem to be the case. So this is the preliminary quantification of that. Again, I think in both cases, it's clearly different. In our TBI case, that we're having overall many more neurons that are active. That sort of correlates with our hyperexcitable network. And then when we're trying to look at differences between neurons next to satellite microglia versus not, we're maybe seeing a little bit of and again, this is all preliminary, so we'll see how this shows up, but we're seeing maybe a little bit of a trend for the satellite microglia to have less percent CFOS positive neurons compared to the neurons that are not next to satellite microglia. So maybe a little bit of a correlate that what we're seeing in our mouse tissue actually does apply to human tissues. And we're trying to also go into some culture microglia and human co-culture models to look more into this. So here's our working hypothesis. We think that satellite microglia exist in the network and sort of wrap around neurons and seem to be important in controlling excitability. And then after traumatic brain injury, they're losing this in some way. And we're trying to understand, well, why is this the case? And if we restore this, could we restore maybe the network after traumatic brain injury and lead to a possible therapeutic option for traumatic brain injury? So some of the things we're working on now is to try to figure out what this protein might be that's important in an interaction between these satellite microglia and neurons. So one of the first things we're looking at has been published a lot about in microglia a lot is the P2Y12 receptor. So this is a purinergic receptor, and there's some really wonderful papers out there that have described that these P2Y12 receptors are really necessary for microglial interaction, particularly at the soma, not so much at the dendrites and the synapses, but really important at the soma and wrapping around the neuron. So we thought, well, that's interesting. Obviously, we probably need a lot of these for our satellite microglia to make contact and wrap around the neurons. And people have shown that there's been more P2Y12 expression, particularly in the satellite microglia. So we have started looking at this in our model and found, interestingly, chronically after traumatic brain injury in the orbital frontal cortex, we do have a reduction of the P2Y12 receptor expression. And this seems to be more likely than other sort of homeostatic microglial receptors. So here's some of the others. And people think that maybe after TBI, you lose the homeostatic signature of microglia. But at least at this 1 to 2 month period, we're finding that these ones, at least in our preliminary studies, are not as affected, but the P2Y12 receptor is as strikingly affected. So we went and did some preliminary staining, and we found that indeed in our microglia that we have a reduction, particularly in the satellite microglia in the soma intensity after TBI, but not so much in the non-satellite microglia. So we're starting to do preliminary work to try to understand if we mess with this receptor, if we can reproduce some of the results we see after TBI. And indeed, when you apply this P2Y12 receptor antagonist into your excitable solution here, we're more likely to see the onset of synchronized activity earlier. And when we apply it, we might see restored excitability in these neurons next to microglia, but this is only an N of 1. So this is all very preliminary, and we have a lot more work to try to do to understand what's going on and how these satellite microglia might be changing neuronal activity. So that's sort of the first part. Any questions here? All right, I'll keep going then. So the next part, talking a little bit about microglia, I'll be focusing in a different region of the brain on the thalamus. So the thalamus, I don't know if any of you are in Murray Sherman's lab. If Murray Sherman even still have a lab. I think probably does, because I feel like he's probably also never going to retire. But maybe Dr. Sherman is right, and the thalamus really is, the most important part of the brain. We certainly see evidence for that in TBI. I know I was very focused on the orbital frontal cortex, but the thalamus is actually another region that we're actually seeing changes that are quite striking in human TBI. So these are 8 patients. They've all been given a TSPO PET ligand, and that's supposed to bind to people think it's activated microglia. They might be astrocytes too. It's not totally sure. But all of these are years to decades after TBI, and every single TBI patient here has increased TSPO binding in the thalamus much more than a control case. So there's something changing in the thalamus with neuroinflammation in general. And people think that might be important in some of the processing or even the seizure production and things like that that occur after TBI. And of course, the thalamus is really important, has sort of connecting all the regions of the brain. And here's just an example of why people think we might be getting more inflammation there. It's when you have sort of axonal injury, perhaps the activated microglia are sort of following the signal of where those broken axons are and end up in the thalamus. I think that's complete hypothesis, we're not sure, but it's certainly an area that has a lot more inflammation, in addition to areas like the orbital frontal cortex. So when I first started here at the University of Washington, the building I'm putting my lab together in actually had someone dropped a cesium radiator. And so they had to like go through and tear out the whole inside of the building. So I couldn't put my lab together. So I started working with a neurologist at the VA who has an electrophysiology lab. And he's really interested in tonic GABA activity after TBI and its involvement in epilepsy. So we decided to look at tonic GABA receptors in the thalamus. And to remind, I'm sure you guys are probably all aware of this, but in case not, GABA receptors are, of course, involved in phasic inhibition in inhibitory postsynaptic currents. But then if they have the delta receptor subunit, they are usually farther away from the synapses and can be activated by GABA and leads to sort of a tonic change in activity. So here's what we decided to do. And there hasn't been really much work done with recording in the thalamus, especially after TBI. So we decided to go look in the medial dorsal thalamus. This area is really important in connecting to the hippocampus and to the orbital frontal cortex. And we first wanted to make sure we're recording from thalamocortical neurons. So this is just an example of their unique firing properties. When they're more hyperpolarized, they have sort of this bursting mode, versus when they're more depolarized, they have this tonic spiking mode. And I'm sure many of you are in neuroscience, you're taking a lot of classes about the importance of this burst and tonic firing mode in sleep and all sorts of network-wide states. So we patched onto these neurons and in voltage clamp, and then decided to do something very simple to look at the level of tonic GABA A activation. We applied different levels. You can either look at this at baseline or you can apply a drug. So we use THIP, which is the delta receptor GABA agonist. And then you spritz on a little bit of bicuculin to block whatever that effect is. And the amplitude here is sort of a measure of how the response for your tonic GABA A receptor response. So surprisingly, we tried this out in the thalamus, and we immediately saw quite a large effect in that we have a much lower amplitude response with increasing agonist to that delta receptor subunit with TBI than we do with our sham. Now, I told you that's very interesting, but this is an area where we have a lot of microgliosis going on. So here's just some stains to show you what that looks like. So here's control of the medial dorsal thalamus. Again, we have our red is Nissl to look at neurons. Green is our microglia. And I think you can start to see these are more branched and thin. And these are getting like a little bit thicker processes here after our TBI. And this is about 4 weeks afterwards. And then our blue here, we're not looking at CFOS anymore. We're actually looking at the delta GABA receptor subunit expression. So we noticed, of course, we have decreased delta GABA receptor expression after TBI. But we also noticed that maybe there was a little bit of an interaction with, microglial interaction. And if you look closely here, we often noticed, and this is just qualitative at this point, we haven't quantified it, but we're seeing especially neurons that have a lot of processes touching them. These might even be considered satellite microglia again. These neurons in particular showed this even more of a reduction in the delta subunit of the GABA A receptor. So we thought, well, this might suggest that microglia are directly involved in some way, of which we don't know, in changing the tonic GABA response after TBI. So to look into that, we decided, well, let's ablate microglia and see what happens. And that's one nice tool that we have in the microglia research world, is we have this drug, which is the CSF1 receptor antagonist, and microglia to stay alive need constant stimulation of the CSF1 receptor, and if they don't have it, they'll disappear. So you can give mice this drug in their chow for 2 weeks and completely ablate microglia. So we gave that to them 2 weeks prior, performed the TBI surgery, and then we continued it until the time of recordings. And you can see there's absolutely no green, no microglia at all after we give them this Plex drug. And when we look at the tonic, when we look at the expression of that delta subunit, it seems to be improved much more in our TBI case after treating them with this. So then we look at the functional response. And indeed, we see that mice that have a TBI but are treated with Plex no longer show any differences in the response to THIP at these different concentrations. So we don't really know exactly how this is happening, and we have a lot more to think about, but microglia seem to be important not only in intrinsic excitability and the production of action potentials, but we're also seeing that they're involved with somehow regulating tonic GABA after TBI. And we have a lot more work to do to try to understand how that is happening. So if anyone is interested in studying microglia and living in Seattle, we're always taking great people from the University of Chicago, so let me know. But those are the 2 main projects we're working on right now. So any questions before we dive a little bit more into the time series analysis that goes into this? Yes, Dana, I see a hand up.
Yeah, I have a question more so about the first half, or the first part so far. I was wondering if you've looked at how the number of satellite microglia or the excitability changes compared to the time since the injury. Like, does it get worse or better over time?
Yeah, that's a good question. We haven't yet looked at how it changes over time. In a different region of the brain, we already see an increase in satellite microglia at 1 week after, but we haven't done recordings to see if the same relationship is there. So I think that will be important to know if this is something that's happening much later on or if it changes from the beginning. So yeah, great question.
Thanks.
Okay, so now I'll get a little bit more into the details of the analysis and focusing on how we actually look at these action potentials. And this is, I think, a pretty basic but important way of doing some time series analysis that you guys have talked about in Ben's class. So to do that, let me open up MATLAB here. Now, again, I want to emphasize, probably many of you in the class can code much better than this, so don't judge me too harshly. But I think it's nice to just go through and especially for those who come from maybe less of a math background like I did, sort of like how you might approach things like this and just some of the little details that are important. And there's also probably, like, if you do a lot of recordings in pClamp, ClampFit can probably do a lot of this for you, but this is actually the same code I used in graduate school that's been tweaked as I've gone back and asked different questions. So keep that code that you guys use. You never know a decade later when you might be using it again. So here is the basic code that I've used to sort of look at all those different action potential algorithms. Just going through here, first there is a file that reads it in and converts it from a MATLAB file into a file that, the ABF file that you get from ClampX into a MATLAB file. And that just came online. That was easy to get. And then I have a step file, and this is essentially, the same data that we're looking at here. So we have a series of hyperpolarizing and then depolarizing current steps that are 250 milliseconds long. And then at some point, when we depolarize the neuron enough, we will start to get action potentials. So that is this step file that we're bringing in here. We define our time axis, which of course needs to represent your sampling frequency. So this is built off of a 20 kilohertz sampling frequency. We're doing sort of a peak or amplitude detection in that we're defining everything to be an action potential if it crosses the 0 millivolt mark. And that could change, but I've set that here, that we have to have that peak cross 0, and then we would analyze it for being an action potential. And the other thing I do, which might be different, you always have to read each paper carefully for how they do things, is I've always looked at action potentials that occur 100 picoamps above where you start getting action potentials. So you're above that threshold, and that's the threshold I'm always using. Different papers use different thresholds. So some use the sort of first current that you get action potentials produced. But in our case, we wanted several action potentials, and we always just use this 100 picoamps above when you start getting that first action potential response. And that's defined just from looking at the steps ahead of time, and then you can change whatever this step number needs to be to match that response. And then our current amplitude is given here. We do a bunch of steps from negative 250 millivolts in 50 millivolt increments all the way up to 700 millivolts. So the next thing is we have our file, we read it in, and it's simply, predominantly, we're focusing on that one step, 100 picoamps above threshold. And we put it through a low pass filter, which I think you guys have a whole lecture talking about filters, because we have to reduce the effect of noise to make sure the analysis works correctly. And we'll get into that a little bit more, but one of the things we do to find the action potential threshold is look at the third derivative. And if we don't filter it first, there's so much little bit of noise in there that the third derivative reading will be abnormally high and not actually find the correct action potential threshold. So that's our low pass filter here. And then after that, we have many different steps here. The first one is relatively easy. First, we go through and we use our spike threshold defined at 0, and we go through and just find, it's just telling the program to go through and find where is that point where we cross the 0. So on one side, we have it above the threshold. On the other side, it's right below the threshold. So we're finding all the 0 crossings here. And those are defined as the spike times. Once we have the spike times, we go through and define the interval between each of them to calculate the adaptation index. So the adaptation index is simply defined as the interval of the last 2 spikes divided by the first 2 spikes. And it's, pyramidal neurons, it's very normal that adaptation rate is above 1 because you have adaptation with regular spiking that occurs over time. In inhibitory neurons, we actually see, especially the fast spiking ones, the adaptation index is much closer to 1 because they have little adaptation or slowing down of the spikes over time. So that's pretty straightforward here. Now, after that, we have the adaptation index. We want to go and determine sort of exactly where we have sort of where we're crossing this threshold on the upside, and also where we're crossing that 0 threshold on the downside. And the reason to do that is, you can certainly go through this code, but essentially we use that later for defining different windows that we want to look at to determine the highest amplitude of the spike and to determine the lowest minimum of that after hyperpolarization. So we go through the whole trace and really define where we're crossing that 0 mark spike threshold above and below. And that's what this code here is doing. And then we can find, once we find all that, we can go through in each of those intervals, identify the highest amplitude and also the minimum amplitude. And then these are used later for different calculations that we'll get into. So after we have those amplitudes and those minimums, then the next thing we want to do is really figure out where the action potential threshold is. And people also have used many different methods to define this. I think in the old days, this was also like something visually done by hand. Probably what's going to happen a little bit to my career as a pathologist right now. We identify a lot of tumors visually and looking at patterns that you identify visually. But as the AI field gets better and as analysis gets better, more of that will become probably computational and looking at genetic expression patterns, et cetera. So I think in electrophysiology, a lot of times people are just going through and looking at the trace and being like, well, I think the action potential is starting like right there. But it's much more precise and has much less bias if you can do something quantitatively the same way every time. So writing a program that will do that will take that bias out of your research, which is really important, as we're finding a lot of things in science have been very biased, and there's been a lot of recall of papers because people have bias that they don't even realize. So the more quantitation, the better. So in this case, the third derivative has actually been something that people have found does a really good job of finding sort of that threshold that people, when it's changing just enough that you really start to get that very fast uprise of the action potential. And here's some commands that go through and find the third derivative of the trace in order to find the action potential. And again, we're going through and doing this a lot based off of different windows that we've defined using sort of the upcross index as an area to look through, sort of the minimum beforehand and then that upcross index, and then looking at the third derivative in that region to find where it's highest. And we really want to find a peak sort of right before we sort of hit that spike threshold. And there can be many peaks, so we often use the local peaks to find the max of interest. And a lot of this had to do with sort of looking at the trace and documenting where is the best area. And I'll show a picture of that in just a second. All right, and then here's the code. Every time you're running analysis like this, even though you think it's perfect, it's always good to just look at what things are doing and make sure it's actually spitting out the data that you think it is. So this is just an example of this red star here is the time and point at which it crosses 0, our spike threshold. And here is the time and point at which it crosses 0 on the way down. So, and this was used to define those different windows that we're using for looking at different things. And then this figure here is plotting our trace of spikes, and then also going through and plotting. The black is the third derivative here. And so we're looking at those peaks that are occurring sort of right before we have that crossing of the 0 line. And we want to make sure that peak aligns with where we think the start of the action potential is here. And we want to make sure we're finding that peak right before here. And I can show you what it's like if we take out the noise and how awful this can get. And we want to find it above a certain threshold here. So this aligns to where we think it should, and all is looking pretty well. So we have the adaptation index. We now have the third derivative, which is the local third derivative max right before we cross the spike threshold, which is considered our action potential threshold. So now we can go through and have those two values. When I'm looking at this in real data, I look at usually the very first spike that's produced. So that's the one. And then I'll often average all the spikes in the trace to look at the average across. And I usually publish the average of those across the trace, but it depends on the question you're looking at. You might be really interested more in that first spike or how the spikes change over time. And of course, you could pull that out if you wanted to in this data. And then after that, we're defining the amplitude here, which should be based off that threshold. So the voltage where we cross that threshold up until, the highest voltage of the action potential. And then here we're defining the half amplitude, which is going to be important in going through and finding the half width of the action potential. And after we define that, we can also go through, just like we defined the amplitude, we can define the after hyperpolarization, which is essentially we want from the area where the threshold is all the way to the minimum. So that's how much the spike hyperpolarizes after the action potential here. So then again, we use those same window phenomenon where we're defining different time points, and we're going through and going to be defining the half width based off of that, which is important and the main metric people use for looking at the width of the action potential here. All right, so now we have our adaptation index, we have our half width, we have our amplitude, we have our after hyperpolarization, we have our action potential threshold. And the final two things I always look at are the minimum and maximum slopes. So we want to know how fast does that action potential rise time get, and how slow is it coming on the way down. And so that's with this code here, and essentially using all these different metrics to define those little windows you're going to look through, and then computing the maximum derivative of the voltage trace and finding, that highest rise time and then the highest falling slope. And we always plot and use that to look at as just another index to define the shape of the action potential. And we'll come to the phase plot in a second. So once we have that, that sort of defines all the main action potential properties that I use. And we've only plotted 3 of them here, but regardless, I would always give a table with all those properties as well that we, in every paper that I am looking at intrinsic excitability, we really want to know, so how the spikes change over time, what is their half width, what is their amplitude, what is the voltage where we have the threshold, and then how fast do they go up and how fast do they come down. So all those indices are important to look at, and I think are helpful in trying to understand, well, what are some of the properties that are changing that lead to how the spikes change or become hyper or less excitable over time. And then the other thing we do is we need this data here. So we need to know not just that one, we need to not just look at this one step, 100 picoamps above threshold when we start to see spikes, but we also need to see how the spikes change over time. And so to do that, we just go through each step and we're going to find the spike index. We're going to then calculate the number of action potentials that get produced. We're going to look at the firing frequency, and we chose to look at the first instantaneous firing frequency, and then we can plot those and look at how those signals change over time. So that's the last step in this code, to look at action potentials. And then I have it spit out all the variables I'm interested in. So usually I have it spit out the average. This is the average half width of the action potential, the amplitude, the action potential threshold, the maximum rising slope, the maximum falling slope, and our after hyperpolarization here. And I also have it do it for the first action potential here and look at that for every neuron I record from. And I usually have these figures plotted out at the same time just to make sure everything looks correct. And that the action potential threshold is indeed, since so much code really relies on this being correct, you want to make sure that that is correct in your code. And so, for example, if you have a particularly noisy trace, and you didn't use that butter filter, then it completely changes your measurements here. Partially because it's no longer reading your third derivative correctly at all with the indices that we have it set up here. So it's really important to know the different characteristics of your trace, how much noise there is, and how that aligns with your definition of your program. All right. Now, the last thing I wanted to talk about, and this is what we're just starting to get into a little bit, is the phase plot. And so here's the phase plot. We're plotting the voltage here and the derivative here. And this is just a better way, I think, of looking at the shape of the action potential in a different phase plane. And why might we do this? Well, a lot of people do this in the field who are looking at the axon initial segment. And the thought is that the spike really starts first in the axon initial segment, then spreads to the soma where it gets amplified even more, and then it goes down the dendrites. So you can sometimes see a little higher blip sort of in the very beginning of the action potential, but it's much actually easier to see it in the phase plot. So this part here is thought to be the action potential being produced in the axon initial segment. And then this is the part that's thought to be produced in the soma. So you can sort of see a little bit of a separation here. So why do I care about the axon initial segment? Well, we're just starting to get into this, but there's been studies out there that have shown that the satellite microglia that wrap around neurons, they actually have a process that goes and aligns with the axon initial segment of neurons. So there's a question of whether they have a role in changing the axon initial segment, maybe their ion channel composition or some type of communication at that point, and whether that can change action potential shape. And that might be suggested by the fact that we have a difference in our action potential threshold with neurons that are next to satellite microglia. So that's what we're hoping to look at next. And then we can quantify things like the peak here compared to the peak there. But one thing I run into is our sampling frequency is not high enough. So a lot of the sampling frequency, so far we've done at 20 kilohertz, but people who study the axon initial segment think to really get the precise resolution you really need a 50 kilohertz sampling frequency. So we're going to be going back and doing recordings at this higher sampling rate to really better be able to look at this component of the action potential coming from the axon initial segment and look at how satellite microglia might be involved or not involved with that part of the action potential shape. All right. Hopefully that wasn't too incredibly boring. I know it can be a little hard to just go through someone else's code, but hopefully the basics are there. Anybody have any questions at all about microglia, action potentials, how to analyze action potentials? Any suggestions for how to improve my analysis?
Speak up if you want to ask her a question. Can you hear me, Amber?
Yep, I can hear you.
I kind of had a couple of questions. The thing with this traumatic brain injury, it seems like the microglia and the GABA A receptors do play a role in arranging excitability. What is the overall idea that is happening with traumatic brain injury? Is the development, for instance, of epileptic activity related to maybe a failure of inhibition, or are there any kind of overarching hypotheses as what is happening with the individual neurons and with the network?
Yeah. So I think at least in the post-traumatic epilepsy field and people who think more in a functional circuit way, I do think that there is changes in the excitatory to inhibitory ratios in the network, and that leads to hyperexcitability. And I think that's why there's some cognitive changes. And I think it's, even if you don't go all the way to having epilepsy, I think, and certainly in these mice that have had these TBI surgeries, it depends on the severity of the TBI and where it is. If you have a more severe one over like the parietal cortex close to the hippocampus, people have found that about 30% of those mice will go on to develop epilepsy. And nobody has looked at this frontal model yet, but I imagine it might be a little bit less. But a lot of times when people have done EEGs, they see changes in the EEG, but they don't actually show up on behavior. So I think that there might be a lot of sort of sub-behavioral responses in the networks that are leading to this hyperexcitability, even if it doesn't necessarily lead to epilepsy syndrome. So that's my working hypothesis, is that networks become hyperexcitable after traumatic brain injury. And a lot of what I'm doing is looking at intrinsic excitability and inhibition. I have found that the non-fast spiking, and we think it's the somatostatin subtype of inhibitory neurons, have deficits in the orbital frontal cortex after TBI. And now we're in a different area of the brain. Of course, we need to look at all of these. In one area of the brain, we're seeing that the tonic GABA response is also reduced. So we think we're seeing changes in inhibition. And other studies have found that there's also an increase in excitation with more sprouting and excitatory synapses and things like that. So I think these are all important things that might be changing that lead to a hyperexcitable network.
Thanks. And the other question I had is a little more technical. You kind of determine, if I understand it correctly, you determine the onset of the action potential from the third derivative. What is the reason you go for the third derivative? Because you kind of noticed correctly that it is very noise sensitive, isn't it? The higher you go in your derivation, the more effect you will kind of have. The more you will be bothered by higher frequencies.
Right. So not everyone uses the third derivative. Some people use the second derivative or the first derivative defined in different ways. But I found if I filter and use the third derivative, it usually hits exactly that peak that I identify by eye. And I don't think there's any more of a reason than that. And when I look in the literature, there's a range, and I've just found that the third derivative, especially for inhibitory neurons, seems to find that that change the best. So that's the reason I've used it. But you're right, it can be more sensitive to noise. And so if you're having trouble getting the noise out of your signal easily, then you might not want to do that and might want to use a different definition for your action potential threshold.
Did you also try the fourth derivative?
No, I haven't tried the fourth.
Because since you're just trying out, I think it's kind of a balance between how far you are willing to cut your high frequencies and how many derivatives you're willing to kind of take without basically changing your signal so much that it's hard to relate the onset to the real signal.
Right, exactly.
Any other questions from either here or the people that are joining us online? Then I would like to thank Amber for. Thank you so much, Amber, and it was really great to see you again after quite a while. We should be visiting Chicago every now and then.
Yeah, I'll be there for SFN in October, so hopefully be able to set up a time to stop by. And before I end, I just, oh, go ahead.
I said to say hello to Karin.
All right, will do. And I did want to, I put their pictures along as we went, but I wanted to thank the people who are actually doing the work in the lab. Corinne and Alicia are working with me now, and they're applying for PhD and MD PhD programs soon. Ryan worked with me. He's now an MSTP student down at the University of Miami. So thanks to them. I'm learning how important it is to have like awesome people working for you. So if anyone ever wants to come to the Northwest and is interested in microglia and electrophysiology and our brain banking, always feel free to reach out. Or if you have any career questions, I'm always happy to chat. And if people wouldn't mind, there is an eval link here. It's always great to get feedback and how I could improve on this lecture if I were to give it again in the future. And that should be in the slides that are in the PDF that's uploaded to the website too.
Thank you.
All right, nice to see everybody. Take care.
Bye-bye. Thanks.
Thank you.